Welcome back to the deep dive. Today, we're jumping into a topic that feels, well, it feels like the future, right? Gene therapy.
It really does.
And we need to be clear from the get-go. This isn't about just managing symptoms with, say, a lifelong pill. We're talking about the quest for a true, definitive cure.
And that's the absolute core of it. We're talking about what's called translational medicine.
Right.
Taking a discovery from the lab.
Yep.
From the bench, really, and moving it directly to the patient's bedside. It's about inserting a healthy gene to, correct a faulty one.
That sounds like an incredibly complex surgical process for the cell itself. So what makes a disease a good candidate for something this advanced? What are the prerequisites?
You're basically looking for two main things. First, you have to know exactly which gene is causing the problem, and you need to be able to clone it or synthesize it in a lab.
Okay, so you need the blueprint.
You need the blueprint. And second, the cells you want to treat have to be accessible. You can't fix what you can't reach.
Right, accessibility. So you're targeting specific locations, not just flooding the whole system.
Precisely. And while, the early trials focused on these simple single-gene disorders, the whole landscape has just exploded.
Yeah.
I think it's critical to realize that today, the vast majority of clinical trials, we're talking something like 65%, are actually focused on cancer.
Wow.
Using genetic manipulation to fight tumors.
Okay, let's unpack the core engineering challenge here. DNA is, well, it's large, it's electrically charged, it's not going to just wander into a cell on its own.
Not a chance.
So if we want to sneak this new genetic instruction in, what are the two main strategies?
We basically categorize them by where the work happens. It's either ex vivo or in vivo.
Let's start with ex vivo, outside the body.
Right. Ex vivo means you take cells out of the patient, often blood cells, immune cells. You treat them in the lab, add the therapeutic gene, and then you put them back.
And the huge advantage there is?
No immune rejection. Because the cells came from the patient in the first place, their body recognizes them. It's a huge hurdle, just completely sidestepped.
That makes perfect sense. So conversely, in vivo means we treat the cells right inside the body. What's the major catch there?
The catch is control. The big challenge becomes making sure the therapeutic DNA only goes to the tissues you're targeting. You want it in the liver cells, say, but you absolutely do not want it ending up anywhere else.
And this gets us to the delivery truck, right? The viral vectors. We're essentially hijacking viruses, stripping them down, and using them to carry our therapeutic payload.
That's a great way to put it.
But given the history, which I know we're about to get into, how sure can we be that these things don't just, turn back into a real virus?
That is the perpetual tension in the field, yeah. And it's why the choice of vector is such a high-stakes decision. The safety engineering is just exponentially better now than it was 20 years ago. We have a whole toolkit of different viruses for different jobs.
Okay, so break down the trade-offs for us.
All right, so you've got retroviruses, which includes modified HIV strains. Their big advantage is that they stitch the new DNA right into the host's genome.
So it's permanent.
Potentially, yeah. Great for long-term expression. But, and this is a huge but, that integration is random. We call it insertional mutation. If it lands in the wrong spot, it can turn on a cancer gene or turn off something essential. A lesson learned tragically early on.
And we use adenoviruses too, right?
We did. Adenoviruses can carry really big genes, which is great, but they are just too visible to the immune system. They can trigger a massive, sometimes fatal immune response. So that led to the current star of the show, the adeno-associated virus, or AAV.
Why is AAV so popular?
Well, for one, it's non-pathogenic, it doesn't cause disease in humans, and it usually doesn't trigger a major immune reaction. But the key thing is, it generally doesn't integrate into the host genome.
So wait, if it doesn't integrate, what happens to the therapeutic DNA? Where does it go?
It just kind of hangs out in the nucleus as a little circle of DNA called an episome. Think of it like a sticky note left in the cell's command center. For cells that don't divide, like in the eye or the brain, that note can stay there for a very, very long time.
But if the cell divides, the note gets lost.
It gets thrown out, exactly, which means you might need repeat treatments. And its other main limitation is size. AAV can only carry about 5 kilobases of genetic material.
And then there are lentiviruses, which seem to combine the best features.
They do. Lentiviruses are also derived from HIV, and they can infect non-dividing cells and carry pretty large genes. And while they do integrate, modern lentiviruses seem to be much less likely to insert into those dangerous gene regulatory regions. The risk is just a lot lower.
It sounds like a really powerful toolkit. But we have to talk about where all that optimism, well, where it came crashing down in the field's early days. We had that first amazing success in 1990.
A huge moment. A young girl, Ashanti DeSilva, was treated for ADA-SCID, severe combined immunodeficiency. She basically had no immune system. They used an ex vivo retroviral vector to give her T cells a working copy of the ADA gene. It was a landmark success.
But then things took a very dark turn. The first major tragedy was the death of Jesse Gelsinger in 1999.
Yes. Jesse was in a safety trial, and the researchers used a very high dose of an adenovirus vector. His body just launched a catastrophic immune response against the vector itself. He died of multi-organ failure.
Yeah.
It put the brakes on the entire field.
And just a year after that, the French X-SCID trials seemed to confirm the worst fears about one of those other vectors.
That was the retrovirus. The therapy actually worked. It restored the kids' immune systems. But then 5 of the patients developed leukemia.
Because of that random insertion.
Exactly. The vector had inserted itself right next to a gene called LMO2 and switched it on, turning it into a cancer-causing gene. It led to uncontrolled white blood cell growth. It was a nightmare scenario.
So you've got random integration causing cancer, a massive immune response, size limits, the potential for creating a real virus. How did the field even move past those obstacles?
It forced a total reevaluation, a complete rethink of safety, of dosing, of vector design. We learned to be far more specific, use lower doses, and people moved away from those old adenoviruses and really focused on making AAV and lentiviruses safer.
And that reassessment brings us to today, where we're finally seeing these genuine, incredible breakthroughs. Where are those lessons paying off?
The eye is a perfect example, a form of retinal blindness called LCA. It's often caused by a defect in the RPE65 gene. The eye is immune privileged, the cells don't divide. It's a perfect place for an AAV vector.
So they inject it right under the retina.
They do, in vivo. And the results, especially in children, have been just remarkable. Gaining enough vision to walk around unassisted. It's a perfect case of the right vector for the right target.
And we're seeing similar success in blood disorders, right? Like Hemophilia B.
Absolutely. A single in vivo dose of an AAV vector delivered to the liver cells, which produced the factor IX clotting protein. And 4 of the first 6 patients were able to completely stop their routine infusions. I mean, that's life-changing.
We're even seeing success in areas that seem almost impossible, like neurodegenerative diseases.
Yes. Metachromatic leukodystrophy, or MLD, a fatal disease. Here, they used an ex vivo approach with a lentivirus, the one with a safer integration, to fix the RSA gene in the patient's own stem cells before transplanting them back, and it halted the disease's progression.
This is where it gets really interesting for me. We're moving beyond just adding a healthy gene to override a faulty one. The goal now is a real surgical fix, to actually rewrite the genetic code.
Gene editing. It has created just unprecedented optimism. Before the tool we have now, there were precursors, like ZFNs and TALENs.
Clumsy, I've heard them called.
Very clumsy. They could cut DNA, yes, but they were expensive and slow and often missed. They paved the way, but they weren't the revolution.
And the revolution, the tool that changed everything, is CRISPR-Cas.
CRISPR-Cas is just so much more elegant. It uses a little piece of RNA, a guide RNA, to find the exact spot in the genome you want to edit. Then the Cas9 enzyme acts like a pair of molecular scissors and makes a clean, double-stranded cut.
And the cell's own repair machinery does the rest.
Exactly. It uses a template you provide to patch the break, effectively rewriting the gene. It's faster, cheaper, and far, far more accurate.
And we're already seeing this in cancer therapy.
Oh, immediately. Think of CAR T-cell therapy like Kymriah.
Yeah.
We're already seeing trials where they use CRISPR to edit the PD-1 gene in those T-cells. Tumors shut down T-cells by hitting the PD-1 receptor. So if you just edit out that receptor,
The tumor can't hit the brakes anymore.
the tumor can't hit the brakes. Exactly.
So if gene editing is fixing the DNA blueprint, the other really big modern approach is RNA-based therapeutics, silencing the gene.
Right. This is about shredding the bad instructions before they can be used. You can use antisense oligonucleotides to just physically block the faulty messenger RNA, or you can use a process called RNA interference, RNAi.
Where the cell's own machinery does the shredding.
Precisely. You introduce a double-stranded RNA, an enzyme called dicer, chops it into tiny pieces called sirnas, and those guide a protein complex called RISC to find and destroy the target mRNA.
And the huge clinical success story here is Spinraza for spinal muscular atrophy.
Spinraza is just a brilliant molecular workaround. SMA is caused by a faulty gene, SMN1. Spinraza doesn't touch SMN1. Instead, it targets the backup gene, SMN2, and it corrects a tiny splicing error that was causing the body to ignore it. It forces the cell to use the backup plans, producing the protein that way.
The pace of this is just astounding, but what are the challenges that are still holding us back from these being commonplace cures?
There are still huge practical hurdles. How do you deliver these to every cell in the brain or muscle? We still don't really know what percentage of cells you need to correct to get a cure. And ensuring that expression is long-term, that's still tricky. And of course, most diseases aren't caused by a single gene.
And finally, we have to get into the ethics, because there's a world of difference between treating one person's body and changing the human gene pool forever.
We draw a very bright line there. Somatic gene therapy, which targets body cells and only affects the individual patient, is what's approved. The controversy is all around germ-line therapy.
Targeting sperm, eggs, or embryos.
Yes, because that change is heritable. It gets passed down to future generations without their consent. It is banned globally for a reason. The early experiments using CRISPR on human embryos showed it's just not safe or precise enough. It causes all sorts of unintended mutations.
And then there's the third category, not curing, but enhancing.
Enhancement gene therapy, or gene doping, using genetics to improve things like athletic ability. That's, of course, banned by organizations like WABA. And that whole debate, where medical necessity ends and enhancement begins, is a conversation we have to keep having.
What a journey, from those early tragic setbacks all the way to this new era of incredible precision with CRISPR and RNA therapies.
The technological hurdles are, one by one, being cleared. But the success of these cures raises this really profound question of equity. A therapy called Leber for a rare disorder failed commercially because its price tag was over $1 million.
So the technology worked, but the economics didn't.
The economics didn't. And that leaves us with the critical question. If gene therapy can offer a true, one-time cure for a devastating disease, who decides what that's worth? And who pays for it? Especially when the alternative is a lifetime of incredibly expensive chronic care. That socioeconomic question might be the biggest hurdle the field has left to face.
A critical thought to leave us with. Thank you for taking this deep dive into the source material with us.
My pleasure.
And thank you, the listener, for tuning in. We'll catch you next time.